Hi, I'm Andrew Bradbury. I work at Los Alamos National Labs, where I'm a scientist and group leader. I've been working on antibodies, phage display, for probably 25 years, and all started really when the human genome was sequenced, and I realized that there was going to be need for antibodies against all those human gene products, and most of my career has been dedicated to try and resolve that goal. I think we're at a moment of real change and disruption, actually. Right now you have something like 300 different providers. If you go on any of these antibody databases, you'll find that there are over 2 million different antibodies available. If you pick any particular target, you may find from 50 to a few thousand different antibodies available. And it makes it really, really difficult to choose. You also can't definitely use the same antibody that somebody else has used in a previous experiment. So my sense is that we're moving away from a situation where antibodies are completely undefined as molecular species to a future where they'll be defined by their sequences. However, there's a lot of resistance from the research reagent companies to that idea. There is some work on the generation of antibodies using in vitro methods, so that's phage and yeast display, against different targets. And I think a few papers coming out in the last 2 or 3 years have shown that we can get antibodies that are as good as those that come out of animals using in vitro methods. I think there's more work to be done there, but I think the future is, the implication is that it's possible to get antibodies against these targets using these in vitro methods. The biggest opportunity is if somebody can discover a way of both providing antibody sequences in such a way that they can make money off the antibodies. The problem, and I think it's a valid concern that the antibody manufacturers raise, is that if they release the sequences of their antibodies, then competitors who have not spent all the money to characterize those antibodies can just produce them and essentially are freeloading. So I think the challenge is to find a way to generate the antibodies, publish their sequences, that allows manufacturers to still make money on the whole issue. The biggest challenge is, as I said, I think releasing antibody sequences in such a way that companies can continue to make money. The reason why it's important to release antibody sequences is that in that way, studies can be absolutely reproduced. That is, an antibody that I've used in a study, if the sequence is available, somebody who comes in 10, 20 years' time can reproduce exactly that same study using exactly the same antibody because the sequence is available. It's not just buying an antibody from a catalog that may or may not recognize its target. I don't think there's going to be such a big development. I think this is an evolutionary process, and I think we're going to be moving slowly towards I think what will happen is that first, more antibody manufacturers will be producing recombinant antibodies, but not revealing a sequence. And at some point, I think we will have some way of releasing sequences.